go ahead and get started. So today we've got a bit of a longer kind of PowerPoint today, but it should be relatively straightforward. So today we're going to be looking at meiosis. So last week we looked at mitosis, which was able to allow us to produce genetically identical cells. Okay. And so that's going to be able to help us produce offspring that are, or produce, cells that are going to be genetically identical to the cell that produced it. It's help us produce new muscle cells and new skin cells and new, new eye cells and things like that. Okay. So today we're going to be looking at meiosis, which is going to be a process that's going to be really important for sexual reproduction. Okay, to help us produce offspring. Okay. Is everyone able to see the PowerPoint? Okay.
Yes.
Yeah. Okay. Cool. Perfect. Alrighty. Coolio. Give me 1 second here. Okay. Hold on. A little bit of it. Okay. Here we go. Okay. Yeah. So we're going to be looking at how sexual reproduction is going to take place. And so, if you kind of think about, if you have any siblings, for example, or think about any siblings that you know, most of your siblings, unless you're a twin, are going to look a little bit different from each other, right? There's some siblings that are, going to be taller, some are going to be shorter. They're going to have lots of different types of characteristics. Okay, and so we're going to see how that's actually going to be able to take place, how we're able to have siblings that don't look alike. And so that is going to be due to this process called meiosis here. Okay. So before we jump into that, we've got a quick question here. So how do prokaryotes divide? Okay, do they divide through mitosis, meiosis, binary fission, or fertilization? Yes, perfect. So I'm seeing this answer everywhere. So binary fission, right? So binary fission is going to be how prokaryotic cells are going to divide, right? They're going to kind of be an individual cell, and then eventually they're going to replicate their chromosome within the nucleoid, and then eventually split off into two cells, okay, which is a lot faster than mitosis, okay, and doesn't take as much energy to do so. Okay. So when we're looking at sexual reproduction is a very energy-intensive process. It takes a lot of energy in order to reproduce. So we're going to look at a few organisms, and we're going to kind of look at how exactly that's going to happen. Right. So if we take a look at the peacock, for example, the peacock needs to be able to produce, especially male peacocks, they need to be able to produce very, very bright feathers. Okay. In order to do so, they have to be eating all kinds of different foods and eating the right kinds of foods in order to help produce very healthy and very bright feathers. Okay. Once they produce those very bright feathers, that's going to be able to attract these lady peacocks all over the place and bring all these peacocks to the yard, right? And so that's going to allow this peacock to be able to reproduce. Okay, so it takes a little bit of energy in order to get to that point where he has, very bright feathers. Okay, if we look at the lion, okay, lion's gonna have kind of a similar thing going on. Okay, so in order for a lion to attract mates, his mane, okay, a male lion in this case, his mane has to be very thick and very healthy, right? If Mufasa here doesn't have a very nice mane, if his mane is all grubby and it looks like he just kind of woke up in the morning and rolled out of the den and was like looking for a mate, okay, that's not going to fly with all the lady lions. And so that's he's going to have a really hard time having some mates. Okay, so again, in order to produce that very healthy mane, Mufasa here needs to be able to eat lots of different types of foods and keep himself healthy. Okay, which is a very difficult thing to do when you're living in the prairie. So very kind of energy intensive kind of process here in order to help him attract mates as well. Okay. Now we look at our male and female humans, right? This process is going to be very difficult too. And so we're going to kind of take things from both perspectives. Okay. I'll talk about the guys first. Okay. So when we're guys are trying to attract mates, ladies, you're going to get a little masterclass in in terms of what's going on in a guy's daily life here. And so for males, when they want to attract mates, there are certain things that we got to do in order to, Kimberly says, spill it, yeah. So certain things we got to do in order to attract the ladies, right? Number one, we got to get out of bed and, shower first thing in the morning. Okay? Some of us on that quarantine lifestyle are not doing that right now, so shame on you. Okay? We got to shower first, okay? We got to shower, we got to put on some deodorant in the morning, and we got to, make ourselves, look decent, okay? Make ourselves kind of look good, right? Look good, do our hair, make sure it doesn't look like we just rolled out of bed. Right, we gotta, there's certain things we gotta do. Okay, Then eventually we got to find the lady. Okay, in this case, find the lady over here. And once we find the lady, then we say, hey, I kind of like you. And then it takes a little bit of energy, a little bit of courage to actually, ask her out. Okay? Then if she happens to say yes, we're not yet in the clear, but we got to now be in the moment where we're not screwing things up. Okay. So there's certain things that we got to do, and this is going to take a lot of energy. There's going to be certain things that we're going to do to start to impress the lady, like maybe start taking on new skills. Maybe we start cooking a little bit more. Maybe we start, taking her out on dates more, and all these different things in order to keep her around, attract her. Okay. So life's a little bit difficult for the guys here. Okay, ladies, we don't have it easy. It's a little challenging on our part. Okay, things not as easy for the ladies. Okay, this is a very energy intensive process here for the ladies too. Now, I'm obviously not a lady, but I am married, so I have kind of a little insight on kind of what goes on here. Okay, so when ladies are trying to attract a mate, guys, again, this is your masterclass here. Okay, they're going to be doing certain things to attract the men. Okay, they're going to be getting up in the morning. They're going to do their hair. Sometimes they'll do their makeup. Sometimes they're going to laugh at your jokes extra hard, even though they're dumb. They're going to bear with you, even though we do dumb things. Things, and then that's not even talking about the hormonal issues that they go through every month. Okay. So and then again, they're going to be doing certain things to kind of keep us around too, and kind of keep us engaged in the relationship. Okay? So, and that's all before we even get to the whole pregnancy point, right? So you can see there's a lot of energy that's going to be expended in order to attract a mate, okay, across the board. Everyone kind of following me so far? Okay. So when we're looking at sexual reproduction, as we can see, it's very, very difficult, and it's Mario's already tired. It's a very difficult process, and so in order to, or kind of, if we take a step back here, if sexual reproduction takes so much energy to do, why has it lasted so long? Okay. Why has sexual reproduction been something that's lasted millions or billions of years, and we still do it today, even though it takes a ton of energy? Why not just asexually reproduce like a bacterium does and just produce clones of ourselves? Why go through this whole process of sexual reproduction? Anyone have an idea why? Why would we do this if it takes so much energy to do? Yeah, so I'm seeing a lot of variation. So it creates genetic variation. Okay, why would that genetic variation be important? Anyone have an idea? And not just for humans, but across the board. So Jared says for evolution, right? So it could be important for evolution. So if we're living in a particular environment, okay, it's going to be difficult if we're just producing clones when we're living in an environment that's not appropriate, not adequate for us, right? If we're in an environment that's way too hot, or an environment that's, there's a lot of pollen in the air, right? It's going to be difficult for us to, live there. Okay? And if we start producing clones of ourselves that, just continue to be the same as ourselves, they're going to also struggle in that same environment. So adding that genetic variation allows offspring to be able to adapt a little bit better to their surroundings, okay? And not just to their surroundings, but to their environment and to different things that are also going on in their environment. So helps with adaptation as a whole, and then also when you take adaptations and multiple adaptations, it can help with evolution of that particular species as well. Okay, so sexual reproduction, even though it's very energy intensive, is a really important process for life to be able to take place and occur. Okay. So we're going to be taking a look at how sexual reproduction is going to be working here pretty much for the remainder of the semester. And so remember, for sexual reproduction, there's two processes that are going to be important. Right? We're going to be looking at fertilization, and that's going to be where we take our two gametes, our sperm and our egg cell, each of those being haploid, okay, one set of DNA or one set of chromosomes within them. Okay? They will then fuse through after sexual intercourse happens, and they're going to form a diploid cell. Then that diploid cell will start undergoing mitosis and then produce your diploid organism. Now, in order to continue going through sexual reproduction, okay, the diploid organism that we produce after fertilization then needs to produce its own or needs to be able to reduce its set of chromosomes from 2 sets down to 1 set. Okay? We need to be able to essentially reproduce the sperm cells and the egg cell. Okay? And so this is going to be done through a process called meiosis. Okay? So in meiosis, there's going to be specific cells that are responsible for building sperm cells and building egg cells. Okay? In order to do that, you're going to have to take that diploid cell, okay, take those 2 sets of chromosomes, and then separate them into having just a single set of chromosomes. Okay? And then that will help us form our haploid sperm and our haploid egg. Okay? So that's going to be the whole goal of this process. The goal of meiosis is to help us produce gametes, help us produce our sperm cells and our egg cells. Okay? And so meiosis is going to look very, very similar to mitosis. In fact, we're going to see many of the same stages in mitosis that we see in meiosis, but they're going to look a little bit different here. Okay? So instead of seeing just one round of division in mitosis, like we see in meiosis that we see in mitosis, okay, we're going to see 2 rounds of nuclear division here. Okay? And so we're going to divide that into 2 kind of subphases of meiosis. We're going to have meiosis 1, and then we're going to have meiosis 2. Okay? So what's going to be happening during meiosis is we're first going to replicate our chromosomes, okay, just like we normally would through like we would in mitosis, and then we're going to divide that cell twice, through meiosis 1 and then meiosis 2. Okay, so we're going to take a look at all the steps that are going to be happening during meiosis. Okay, so we're going to start with meiosis 1, because that's going to be the first, the first of the 2 processes here. But before meiosis 1 even hits, we're going to have an interphase, just like we had during the mitosis. Okay? So we're going to have an interphase, and we're going to have the same phases within interphase. We're going to have the G1 phase, the S phase, and the G2 phase, and they're still going to have the same checkpoints during each of those stages. Okay? Now in S phase, just to kind of remind you, in S phase, we're going to be replicating the chromosomes. And so that's going to help us to form our 2 sister chromatids here. So we're going to have one chromatid on this side, another chromatid on this side, and they're going to be linked at the centromere. Okay? In addition to that, we're also going to replicate the centrosomes, okay? Just like we did in mitosis. Nothing different is happening here. Okay, same process. Okay. Okay, so before we get into meiosis, what is the role of the centrosome? Does the centrosome act as the attachment point for the microtubules? Is it the attachment point of the sister chromatids? Does it cause the formation of the cleavage furrow? Or is it just for decoration? What do we think? Kimberly says D. So the answer here, and a few people are getting this, is A, right? So it's going to act as the attachment point for the microtubules. So the centrosomes are going to be producing those microtubules that will become the mitotic spindles. And those mitotic spindles will eventually attach to the, in order to attach to the chromosomes. Okay. All right, so the goal of meiosis is going to be different from mitosis, okay? So in mitosis, we were trying to produce genetically identical daughter cells, okay? In meiosis, we don't want to be producing genetically identical daughter cells, okay? What we want to do is we want to produce daughter cells that are genetically unique to the parent cell. And when the daughter cells are genetically unique, that is going to allow the offspring to be unique and different from the parents that produce them, okay? So we want to produce genetically unique cells during meiosis, okay? And that's going to help us to produce variation within our offspring. The genetic uniqueness within those daughter cells is what's going to allow your siblings to look different from one another and have different traits from one another. Okay? And that's also going to allow variation across the entire species, okay? Because everyone's producing genetically unique cells, everyone is going to look a little bit different, okay? Or in some cases, very different from one another. So there's going to be 2 places in meiosis, or 2 specific events in meiosis that are going to contribute to genetic variation. And we're going to take a look at those as we go here. Okay, so the first phase of meiosis 1, okay, again, we're starting with meiosis 1 here, is called prophase 1. Okay? And so in prophase 1, there's a very specific event that's called crossing over. And crossing over is the first place where we see genetic variation begin to occur. Okay? So what's going to happen in prophase 1 is we're going to break down the nuclear envelope, okay, just like we would in mitosis. We need to be able to access those chromosomes. Okay, so we're going to break down the nuclear envelope, and then we're going to take our homologous pairs of chromosomes. For example, we're going to take, mom's, or the chromosome number 4 from mom, and then chromosome number 4 from dad. Those are going to be homologous chromosomes, and they're going to come into close alignment with one another. Okay? So when they're brought together, okay, that's the process called synapsis. Okay? Synapsis is just when you're bringing those homologous chromosomes together. Okay? Now remember, those homologous chromosomes are going to have the exact same genes in the exact same locations. Okay, so for example, we're looking at, this might be chromosome number 4 that came from mom, okay, or that came from dad here. Okay, we have gene A over here, we have gene B down here, and gene C down here. Okay, when we look at mom's version of the homologous chromosomes, okay, again, we're gonna have gene A, gene B, and gene C. Okay, so remember, in our organisms here, okay, we're starting with a diploid organism, a cell that has 2 sets of chromosomes, one that came from mom, one that came from dad. Okay, so this is your paternal chromosome, and this is the maternal chromosome. Okay, so the first thing that's going to happen is they're going to come into close alignment with one another. Okay, and they're going to come in such close alignment with one another that even their genes are going to be very precisely aligned with each other. Okay, they need to be very close in alignment with one another in order to make this work properly. Okay, so what's going to happen is each of these chromosomes is going to kind of cross over onto the other one. Okay, so you're going to have one sister chromatid from the maternal chromosome, and that is going to cross over a sister chromatid from the maternal chromosome, okay? And they're going to link across each other. Okay? The point where they cross is called the chiasma. Okay? So the sister chromatids are literally going to actually cross over one another, and they're going to kind of bind and join to each other in that orientation there. Okay? When they bind to each other at the chiasma, this is going to be a point where they're going to exchange their DNA with one another. And so a part of this paternal chromosome is going to get exchanged with the maternal chromosome. And so what's going to happen is we're going to have a set of homologous chromosomes that now look like this, okay, where they're literally going to exchange DNA with one another. Okay? So we call this now having our recombinant chromosomes. Okay? So this picture is a little bit slightly inaccurate here, in that here we do see that the that crossing over has taken place, where a little bit of the DNA from mom has crossed over onto the chromosome from dad, and a little bit of the DNA from dad has crossed over onto the DNA from, or the chromosome from mom. Okay, however, these are still going to be linked at the chiasma. Okay, they're not going to be separate at this point. They're still going to be linked here. Okay? And so as they remain attached to each other at the chiasma, the homologous chromosomes are going to continue to condense here. Okay, and so now because they're all linked together, okay, now we have our homologous chromosomes linked together. We now refer to this pairing as a tetrad. Okay? A tetrad meaning 4. We have 4 sister chromatids that look different from one another. We have one that's all blue, one that's got mostly blue and a little bit of red, one that's mostly red with a little bit of blue, and one that's completely red. Okay? So each of these sister chromatids looks completely different from the next one. And so this is going to be the first place where we see genetic variation begin to occur. Okay? And so we call these sister chromatids that had the crossing over occur recombinant chromosomes because they combine their DNA with one another. Okay? So these would be the recombinant sister chromatids here, okay? This leg over here and this leg over here, while the ones that did not have any crossing over occur are called non-recombinant chromosomes or non-recombinant sister chromatids. Okay? So that is crossing over. That's going to be the first event where we see genetic variation occur in meiosis. Okay? So again, initially, we start off with 2 homologous chromosomes. Remember, they have to be corresponding chromosomes. So chromosome 4 will always line up with chromosome 4. Chromosome 1 will always line up with chromosome 1. But you'll never have, for example, chromosome 4 lining up with chromosome 1. They're not identical cells. And so, or they're not corresponding cells. They're not identical. So they're not going to be able to link up with each other. Okay, so these are going to be between corresponding chromosomes here. Okay, they're going to link up. Okay, they're going to align themselves appropriately. And then they're going to cross at the chiasma. Okay. Once they cross, they're going to exchange some of their DNA, and now we have recombinant chromosomes. Okay. Any questions on crossing over here first? If this makes sense, go ahead and throw a check mark or a star, or tell me it makes sense in the chat or something. If it doesn't, go ahead and throw an X or have me go through this one more time. We feel okay with this. Cool. Okay. Cool. All right. So that is crossing over. So that's going to happen during prophase 1. Okay. And the next parts of prophase 1 are going to be exactly the same things we've seen previously. Okay. So in prophase 1, what we're going to see is we're going to have our spindle fibers, our mitotic spindles, coming from the centrosomes, and they're going to align, or they're going to attach to the chromosomes at the kinetochores, which are at your centromeres, okay, just like they normally would. Okay. So your sister chromatids, or sorry, your spindle fibers, your mitotic spindles are going to attach to the kinetochores. Okay. And they're going to make sure that they are fully attached, and the chromosomes, the centrosomes are going to start moving to opposite ends of the cells. Okay. Now remember here, the chromosomes are still attached at the chiasma. Okay. One thing that I forgot to mention previously is that even though in the initial picture I showed you, crossing over happened in just one location, that doesn't actually happen in just one spot. Usually crossing over will happen in multiple points along the chromatids. Okay. And sometimes it can be in tens, and sometimes it can be hundreds of locations where you're going to have crossing over take place. It's not going to be just one spot. And so here, for example, this picture isn't quite accurate from what we've seen already, but you might have crossing over happen over here, and you might have crossing over happen over here as well, okay, and other points across the chromosomes. Okay. So remember, your homologous chromosomes are still going to be attached at the chiasma, even though even after the kinetochores have now attached to their mitotic spindles. Okay. And then the last thing that's going to happen is the nuclear envelope is going to fully break down. Okay. So we have the same events essentially that happen in prophase of mitosis. The only difference here is that we're lining up the corresponding chromosomes and having crossing over occur. Okay, so that's prophase 1. That is the first of the two phases where we see genetic variation begin to develop. Okay. Oops. Okay. Now the second phase is called metaphase 1. Okay. So metaphase 1 is going to be the second place where we see genetic variation occur. Okay. And so what happens in metaphase 1 is we're going to have our chromosomes line up along the metaphase plate. Okay. However, because our chromosomes are still attached at the chiasma, they cannot line up in a single file line. Okay. Instead, what's going to happen is those homologous chromosomes are going to line up on either side of the metaphase plate. They're still going to be lined up along that metaphase plate, but instead of being in a single file line, okay, they're going to be lining up on either side of it. Okay. Now this is called independent assortment because the chromosomes are going to arrange themselves and orient themselves in a random orientations along the metaphase plate. So for example, we might have a maternal chromosome on this side and then a paternal chromosome on the other side. Okay. The next one might have a maternal chromosome on this side and then a paternal chromosome on the next side. Okay. And the next one we have it reversed, where we have the paternal chromosome on the left side with the maternal one on the right side. Okay. So they're going to orient themselves just in any random orientation across the metaphase plate. Okay. You're never going to have all of the maternal chromosomes on one side and all of the paternal chromosomes on the other. They're going to be randomly oriented, okay, randomly assorted along the metaphase plate. Okay. And so their orientation, or the way they're arranged, is going to be independent of any of the other chromosomes. Okay, so there's nothing that's going to regulate how it's going to be oriented. It's going to orient itself however it happens to orient itself. Okay, and so you're going to end up with random numbers of paternal chromosomes on one side and random numbers of maternal chromosomes on the other side. Okay. Okay. Does this one make sense? Okay. How do you feel? Okay. Yeah. So again, here the chromosomes are just lining up randomly across the metaphase plate. Okay, but they are going to be lined up, along each other, not in a single file line, but in pairs. Okay. So that is the second of the two places where we see genetic variation. The first one was in prophase 1, where we saw crossing over. The second place is in metaphase 1, where we see independent assortment of our chromosomes here. Okay. So because all the tetrads orient themselves in all these different types of variations, or all these different types of assortments, that is going to give us a huge variation in the amount of offspring that we're able to develop. Okay? And so because you have 23 chromosomes that are all going to be lining up however they decide to line up, okay, there's over 8 million possibilities, okay, of this genetic variation. Okay, and so for that reason, every sperm cell is going to be completely different from the next one. Each sperm cell is going to have a completely different complement of DNA, okay, than the next one will have. Okay, and again, that's just because of the independent assortment. Okay? And same thing with the egg cells. Okay, egg cells are going to do exactly the same thing. Independent assortment is going to happen, and that's going to give over 8 million possibilities of genetic variation within, you know, other egg cells. Okay. And so that is what and so that's just taking into account independent assortment. Okay, that 8 million possibilities doesn't even take into account, you know, the effect of crossing over. Okay, so as you can see, you can end up with all kinds of different possibilities of you know, complements of your genome, okay, just by prophase, or just by independent assortment and from crossing over. And that is what's going to contribute to the genetic differences between siblings. Okay? Even though they have the same parents, they're going to be producing offspring that look very different from one another because of independent assortment taking place and because of crossing over occurring. Okay? So no haploid cell is going to have the exact same composition as the next one. Okay, so which is incredible, which is completely mind-blowing, that you can create so many different possibilities of offspring just through these two processes of prophase 1 and metaphase 1. Any questions on prophase 1 and metaphase 1 so far? Okay. Okay, so that's the first two steps. Okay. All right. Then pretty much the rest of this is going to look kind of as we normally have seen it. Okay, so the next one is going to be anaphase 1. Okay, so in anaphase 1, we're going to have our spindle fibers attached to the chromosomes, which are already attached at the during metaphase 1. Those spindle fibers, our mitotic spindles here, are going to start to pull apart the chromosomes, and they're going to separate them at the chiasma. Okay, and so we're going to end up breaking apart our chromosomes here. However, the sister chromatids, each individual sister chromatid is still going to remain attached at the centromere. Okay, so all we're doing here is we're breaking apart the homologous chromosomes from the chiasma. Okay, and so this is why independent assortment helps to create such an incredible amount of genetic variation. Once you pull these chromosomes to opposite ends of the cell, okay, now you're going to have, you know, a random assortment of these chromosomes that are now going to be divided up into these two new cells here. So anaphase 1, okay, we're pulling apart our homologous chromosomes at the chiasma. Okay, but the sister chromatids are still remaining attached. Okay. And then the last phase is telophase 1. Okay, so in telophase 1, again, exactly the same things are happening. So our chromosomes are going to end up on opposite poles of the cell. Okay. In some cases, in some species of organisms, the chromosomes will then decondense. And then they're going to reform the nuclear envelope around those chromosomes. Okay? So you end up forming two nuclei, okay, inside of one individual cell. Okay? Again, just like we saw in meiosis. Okay? The only difference here is that we have these full chromosomes, okay, instead of individual sister chromatids that were separated. Okay? So some organisms will decondense the chromosomes and then reform the nuclear envelope. Some won't. It just kind of depends on the species. And then the last phase is we have cytokinesis. Okay? So in cytokinesis, we're going to end up splitting the cell, okay, using either a cleavage furrow if we're looking at animal cells, or cleavage furrow, or the cell plate. Okay? And then we're going to end up forming two daughter cells. Okay? And so that is going to be the end of meiosis 1. Okay? So a question here. So which type of organisms require a cleavage furrow and a cell plate for cytokinesis? So we got A, plants require a cleavage furrow while animals require a cell plate. B, animal cells require a cleavage furrow and plants require a cell plate. C, both animal cells and plant cells require both a cleavage furrow and a cell plate, or both animal and plant cells require only a cleavage furrow. What do we think? Yes, I'm seeing B, and B is correct, right? So animal cells will be producing the cleavage furrow in order to separate the cells. Okay, plant cells will be producing a cell plate, okay, in order to separate the daughter cells. Perfect. Okay. So once we go through this process of cytokinesis, the daughter cells that we produce, those two individual daughter cells, are going to be considered haploid. Okay, so remember at the beginning of meiosis, we had two sets of chromosomes. We had a maternal chromosome on one side and a paternal chromosome on the other side. Okay. During anaphase, you end up separating those chromosomes, okay, and so they end up forming into these new individual cells. Okay. Now, because you only have one set of chromosome in each cell, you only have one chromosome 1, one chromosome 2, one chromosome 3, and so on and so forth in each cell. Now you only have one set of chromosomes in that cell, so we consider that to be haploid. Okay? And same thing with the opposite cell. So you end up going from a diploid cell, okay, which had two sets of chromosomes. You separated those chromosomes to individual cells. Okay? And so now you only have one full set of chromosomes in each of the daughter cells. So they're now considered haploid. Okay? Now again, remember here, the sister chromatids are still going to be attached, and still going to be attached as a result of crossing over. Okay? But they're not going to be identical. Okay? So the chromosomes that you end up forming in each of your cells are not going to be identical because crossing over had already taken place. Okay? So our daughter cells at this point are haploid. Okay. So question here. So which two phases of meiosis 1 directly result in genetic variation? Is this prophase 1, prometaphase 1, metaphase 1, anaphase 1, or telophase 1? Okay, there's two answers for this one. Which ones do we think? Anybody? So Mario's got this one. So, and Yeti too. So the two that result in genetic variation directly, okay, are going to be prophase 1 and metaphase 1. Okay, remember, prophase 1 is going to be having crossing over occur, and that's going to help to contribute to some genetic variation. And then metaphase 1, which is where we're going to have independent assortment. So our chromosomes are going to line up independently, they're going to line up randomly, and then eventually are going to get pulled to opposite ends of the cell. Okay, so the two daughter cells that you produce are going to be completely different from one another because they're going to have a different set of DNA within them. Okay, so those are the two places where we see genetic variation occur. Okay, perfect. Yeah, so crossing over and then independent assortment. Beautiful. Okay, so this is kind of an overview of what meiosis 1 looks like. So we started with prophase 1, where we got rid of our nuclear envelope. Okay, we had some homologous chromosomes start to attach at the chiasma, okay, and then they start undergoing crossing over. Okay. We still form our cytosomes, which will produce our mitotic spindles, and they're going to start moving to opposite poles of the cell. Okay. Then we end up in metaphase 1. So at metaphase 1, we have our mitotic spindles attached to the chromosomes already, and then so they're going to be attached and they're going to line up along the metaphase plate. Okay. In anaphase 1, we're going to break the chiasma and we're going to move each individual chromosome to opposite ends of the cell. Okay. And then telophase 1, we end up forming our new nuclei around each of those cells. And then cytokinesis, we're going to end up splitting those cells into two. Okay. So that's meiosis 1. Any questions on meiosis 1 before we get into meiosis 2? Okay.
So just a small question with telophase 1 and cytokinesis, are they considered separate steps or are they considered one step?
Yeah, so they're considered separate steps. This picture doesn't really show it that well, but they are considered separate steps. Yeah, just like you would see it in mitosis. So telophase 1, they're still going to be within, you're still going to have those two nuclei within that one cell. And then you go through cytokinesis and you actually split it into two individual cells. Yeah. So Yetzi's question is, so is there any way to predict the type of genetic variation an individual will have before it's born? No. So there's no way to predict it. And that's the reason you can't predict it is because, again, you're having 23 chromosomes all go through crossing over, okay? And they're all going to be going through crossing over in random places, okay? So there's no way to predict that point. And then because you have 23 pairs of chromosomes all aligning along the metaphase plate, there's no way to predict which side of the metaphase plate they're going to line up on. So there's absolutely no way to predict it. And so that's kind of the fun of, I guess, having babies, I guess. You never know what you're going to get. You never know exactly the complement of genes, or the complement of genes, and what your baby's going to end up looking like through this process here. Yeah, so no way to predict it. It's all a mystery until it happens. Yeah, very good question, Yancy. Any other questions? Okay. All right. So that's meiosis 1. Okay. Now we're going to start going through a second process called meiosis 2. Okay. And so meiosis 2 is where we take those 2 haploid cells that we produce at the end of meiosis 1, okay, and they're going to undergo meiosis 2, and they're going to end up splitting into 4 haploid cells. Okay. And so meiosis 2 is going to look actually very, very closely similar to mitosis. Okay. And so we're going to take a look at what that looks like here in a second. So in some species, there's going to be a very short interphase in between meiosis 1 and meiosis 2. Okay. And we call that very short interphase interkinesis. Okay. Now, in interkinesis, there's not going to be the same events that are happening in interphase, like we saw before meiosis 1. For example, we're not going to see an S phase. Okay. And so we're not going to be replicating our chromosomes. The chromosomes that we produced at the end of meiosis 1 are the chromosomes that we're going to be working with exactly as they are when we enter meiosis 2. Okay. Okay. However, we are going to replicate the centrosomes. Cool. So the centrosomes at the end of telophase 1 ended up breaking down. Okay. So we have to re-replicate our centrosomes in order to help us undergo meiosis 2 properly. Okay. So here we go. So we're going to start with prophase 2. Okay. And so again, prophase 2, we're going to have the same events that we have happening during prophase. Okay. Prophase and then prophase. I'll just leave it at prophase. So in prophase 2, what we have is our chromosomes are going to condense. Okay. So they were initially decondensed at the end of telophase, and they're going to recondense again. Okay. We're going to break down the nuclear envelopes because, again, we want to be able to access those chromosomes. And then the centrosomes that we produce are going to end up moving to opposite ends of the cell. They're going to undergo the production of the mitotic spindles, and those mitotic spindles are going to attach to the kinetochores of each sister chromatin. Okay. However, there is not going to be any new crossing over that's going to be taking place. Okay, so again, these are our chromosomes that we had at the end of meiosis. At the end of meiosis 1. Okay, they already undergone crossing over when they were in prophase 1. Okay. When they enter prophase 2, there's no more crossing over. This is just going to be regular old prophase happening here. Okay. Nothing special is happening. Okay. Okay. Then they start to undergo metaphase 2. Okay, so in metaphase 2, they're actually going to line up along the metaphase plate. Okay. So they're going to line up along the metaphase plate, again, in a single file line this time, okay, because you're only dealing with one set of chromosomes. Okay. So they're going to line up along the metaphase plate. Then we get to anaphase 2, where you pull the sister chromatids apart, and you move them to opposite ends of the cell. Okay. And again, you're going to be using the spindle fibers to do that. Okay. And then the last phase that we'll have is telophase 2. So in telophase 2, the chromosomes end up on either end of the cell. They begin to decondense, and then you reform the nuclear envelopes around each of these cells. Okay. So here's one cell with 2 nuclei in it. Here's another cell with 2 nuclei in it during telophase 2. Okay. At the end of telophase 2, we have cytokinesis, and then we end up splitting each of these cells into 4 genetically unique haploid cells. Okay. These 4 cells that we produce here will become the sperm cell or the egg cell, depending on the sex of the individual. Okay. And so as we can see here, each of these each of these cells is going to have a different chromosome arrangement. Okay. All these chromosomes are going to look very different from one another. Okay. So here we got, full blue and full purple. Here we're going to have a little bit of a different, we're going to have a little bit of blue and a little bit of purple in one chromosome, and a little mostly purple and a little bit of blue on this one. Okay, this chromosome or this set of cells is going to look different, and this cell over here is going to look different. Okay. So remember, at this point, we're still dealing with haploid cells. So our cells are still haploid. They've undergone just a regular old nuclear division here. Okay. And so we end up with a single set of chromosomes. And so these are all still considered haploid cells. Okay. So these are the gametes that we will be producing here. Okay. Okay. Any questions on meiosis 2? Okay, meiosis 2 is very, very similar to mitosis. Okay. Same thing is happening. The only difference here is that our chromosomes have already gone through crossing over, so they just look a little bit funky, okay, than they would ordinarily. Okay. Any questions on meiosis 2? Okay. Okay, so this is what this process would look like, kind of as an overview. Again, dealing with starting with prophase 2, you have chromosomes just kind of oriented along the cell. They're going to be attached to the mitotic spindles. In metaphase 2, they're going to line up along the metaphase plate. In anaphase 2, we separate them at the sister chromatids. Okay. And then we end up with telophase 2, and then cytokinesis to split them up. So Kimberly's question is, why are some shorter? They shouldn't be shorter. And this was just kind of not a very good drawing, but they should be about the same length. Okay? They shouldn't be shorter because you're still dealing with the same chromosomes. Yeah, this is just a bad picture, but it kind of emphasized what I was talking about as far as what was happening within the cells. Yeah. Any other questions here? Okay, so that's meiosis 2. Okay. So if we were to take a look at meiosis 1 and leading into meiosis 2, again, you would start off with having your prophase 1, where you have your homologous chromosomes attached at the chiasma. Okay. They're going to undergo crossing over. We break down our nuclear envelope. We form our centrosomes, which will be producing our mitotic spindles. And then we enter metaphase 1, where our homologous chromosomes, our tetrads, are going to be lining up on either side of the metaphase plate. Okay. Anaphase 1, we pull each individual chromosome apart, okay, to opposite ends of the cell. And then telophase 1 and cytokinesis, you end up reforming the nuclei around those chromosomes and then splitting that cell into 2 individual cells. And then you enter meiosis 2, where you have prophase 2. Okay. And so you end up, again, forming your mitotic spindles again. Metaphase 2, they're going to line up along the metaphase plate in a single file line this time. Okay. Anaphase 2, you're going to separate them by their sister chromatids. Okay. So the sister chromatids are going to get moved to opposite ends of the cell. Okay. And then telophase 2 and cytokinesis, you're going to end up reforming the nuclei and then splitting the cell into 2. Okay. Is this all making sense? If it's making sense, go ahead and throw a check mark or a star. If there's some confusion, throw an X. Or, if there's a question, let me know. How do we feel about meiosis 1 and meiosis 2? Okay, cool. Okay, looks like we're feeling good here. Okay, so again, the goal for meiosis 2 is to produce genetically unique daughter cells. Okay, and so when we produce those genetically unique daughter cells, that is going to give us the variation that we see between siblings, okay, and across our species as a whole. Okay, so when we're looking at meiosis, there's going to be a few similarities. Oops, sorry. There's going to be a few similarities and a few differences between mitosis and meiosis. Okay, so we're going to kind of compare the two. And so when we look at mitosis, remember the goal here was to undergo just one single nuclear division to produce 2 daughter cells. And those daughter cells are going to be genetically unique, and they're going to have the same number of chromosomes as the parent cell did. Okay. In meiosis, we're going to have 2 nuclear divisions. Okay. So we're going to divide twice, and we're going to end up forming 4 cells. Okay. And each of the cells that we produce is going to be haploid, meaning that they're only going to contain 1 set of chromosomes. Okay. And each of these cells that we produce is going to be genetically unique to each other and genetically unique from the parent, okay, or from the parent cell. Okay. So there's going to be complete uniqueness across the board in meiosis, whereas in mitosis, we're essentially producing genetically identical cells. All right, so Mario has a question. So Mario says, so it seems like this is just making a copy of a copy of a copy. Does it ever create problems? So yes and no. So it's not necessarily making a copy of a copy and copying that copy. It's kind of changing things around, right? It's causing crossing over to take place. It's causing independent assortment to take place. And so you're not essentially developing copies of cells. However, this does create there are some problems that can arise, and we're actually going to talk about some of those problems in this next section that we get into. So there are some problems that can arise through meiosis if something does not work properly. Cool. All right, so that is mitosis and meiosis. Okay, so then a few other differences. So in mitosis, again, when we're looking at mitosis, our chromosomes are going to line up in a single file line along the metaphase plate. Okay. However, in meiosis 1, okay, if we're looking specifically at meiosis 1, we're going to be looking at homologous chromosome pairs instead of individual chromosomes in mitosis. So what's going to happen is those homologous chromosome pairs are going to be bound together at the chiasma because of crossing over, and they're going to line up on either side across the metaphase plate in their tetrads. Okay. So that's going to be a difference in metaphase between mitosis and meiosis 1. Okay. Then in anaphase, in mitosis, we separate the sister chromatids themselves, okay, and they get pulled in either direction. In anaphase 1 of meiosis 1, individual homologous chromosomes are pulled apart, okay, in either direction. Okay. And the last thing that's a difference here is that in mitosis, we have what's called no reduction division, essentially meaning that the number of chromosomes that we have, or the number of sets of chromosomes that we have, is going to be exactly the same between the parent cell and the daughter cell. Okay. However, in meiosis 1, we do have a reduction division, which means that we're reducing our sets of chromosomes from 2 sets of chromosomes to now 1 set of chromosomes. Okay. So meiosis 1 has a reduction division. Mitosis does not have a reduction division because you're ending up with the same number of chromosomes in the daughter cells as you did in the parent cell. Okay. All right. Now, if we compare mitosis and meiosis 2, we're going to see similar trends here happening. So one difference, though, is that in mitosis, the daughter cells that you produce are, again, going to be genetically identical to the parent cell that produced it. Okay. In meiosis 2, the daughter cells are all going to be genetically unique to each other, and they're going to be genetically unique to the cells that produce them because of crossing over and because of independent assortment. Okay. However, these are both not going to be reduction divisions, okay? Because you're starting meiosis 2 and ending meiosis 2 with the same number of chromosome sets as you had at the beginning. Okay? Same thing in mitosis. Okay? You're ending with the same number of sets of chromosomes as you did at the beginning of this process. Okay. All right, so this is a table that's in the textbook that kind of goes through some of the differences between mitosis and meiosis, going over a few different types of processes here. Okay. All right, so question here. So which of the following is true about crossing over? Okay, so A, most homologous pairs do not have any crossover events. B, a chiasma indicates that a crossover has occurred. C, segments of DNA are traded between unrelated chromosomes. D, crossing over occurs more often in male gametes than female gametes. Okay, what do we think here? Yeah, so I'm seeing some B's. Yeah, perfect. So B is correct. Yeah, so crossing over is going to take place at the chiasma. Okay. So the chiasma is going to indicate that a crossover event has happened. A is wrong, right? Because most homologous pairs will have a crossover event. And in fact, every homologous pair does have a crossover event taking place. Okay. In C is wrong because we're not going to have segments of DNA being traded between unrelated chromosomes, right? Only chromosome 4 is going to be able to cross over with chromosome 4, okay, of the of the kind of counterpart chromosome. Okay, you're not going to be able to have chromosome 1 have a crossover with chromosome 20. Okay, it can't happen because they're going to be different. They're going to have different sets of genes on them, and they're going to be different sizes, and so they're not going to be able to align properly. Okay, so crossover is not going to happen between them. And then crossing over is not going to happen more often in males and females. It's going to happen at pretty much the same rate. Okay, next question. So which of the following is true of meiosis 1? Okay, so A, most homologous pairs do not have any crossovers during prophase 1. B, chromosome number is reduced from diploid to haploid. C, sister chromatids in a cell undergoing meiosis are always identical. Or D, sister chromatids separate to opposite ends of the cell. Seeing some B's. And so B is the correct answer. That's perfect, right? So at the end of meiosis 1, remember, we're going from a diploid cell to now having 2 haploid cells at the end of meiosis 1. Okay. Again, A is wrong because, again, most homologous pairs are going to have crossing over in prophase 1. C is wrong because we're not going to be separating sister chromatids. Okay, we're going to be separating chromosomes in meiosis 1. We'll be separating the sister chromatids in meiosis 2. And then the sister chromatids getting separated to opposite poles of the cell. Okay, again, that's not going to be happening here. So yeah, perfect. Okay. Next question. So which phase of meiosis is this? Are we looking at metaphase 1, anaphase 1, metaphase 2, or anaphase 2? What do we think here? Okay, so I'm seeing 2 conflicting answers. Yeah, so the majority has got this one right. So this is anaphase 2. Okay, how do we know this is anaphase 2 and not anaphase 1? Can anyone tell me?
Isn't it because there's just one single line of each of the cells and they're starting to split again?
Right. It's because we're looking at our sister chromatids that have been separated from each other, right? So we do see that crossover, and so that's another way that you can tell too, Paul. So Paul says that crossover has happened. So you can tell that crossing over has already occurred, okay? And we are looking at individual sister chromatids, okay, being moved. If we were looking at anaphase 1, you would have the full chromosome being moved to opposite ends of the cell, okay? However, here, we're just looking at the sister chromatids being separated to opposite ends of the cell, okay? So if we're looking at sister chromatids getting separated, we know we have to be looking at anaphase 2 and not anaphase 1. Cool. Very good, very good. Okay, so that's meiosis. Are there any questions on meiosis here before we move into some of the errors that can happen in meiosis? Any questions? Okay. So there's no questions. We'll go ahead and jump into meiosis and some of these errors that can happen. So meiosis, like we see, is a really important process to help us produce genetically unique cells. Okay. We need to produce genetically unique cells here. However, there are some problems that can take place in meiosis if something doesn't work right in meiosis. If there's some kind of issue that happens at different stages of meiosis, and we can end up with some disorders, and these can lead into various types of genetic disorders. Okay. So we can have 2 main classes of errors that can happen in meiosis. Okay. The first one is called an error in chromosome number, okay? And then the second one is an error in the disorder of chromosome structure. Okay. So errors in chromosome number is where instead of end up forming 2 chromosomes, okay, or having 2 chromosomes in your diploid cells, you end up with either 3 sets of chromosomes, okay, or a set of 3 chromosomes, or 1 chromosome. Okay. So instead of having the 2 that you're supposed to have, you end up with either too much chromosomes or too little chromosomes, okay? Or too few chromosomes. The other thing that you can have is called a disorder of the chromosome structure. Okay? And so that can be something where the chromosome is actually just not built properly. Okay? There's either a piece missing or a piece added, or there's something wrong with that chromosome that's either there that shouldn't be there, or that's not there that should be there. Okay? So we're going to take a look at both of these different processes, and we're going to spend the majority of our time looking at disorders of chromosome number. Okay. So disorders of chromosome number are easily seen using these tools called karyograms. Okay? And so karyograms are really cool because they're able to take a picture of the chromosomes that are happening, or the chromosomes that exist within your cells. Okay? And they're going to be able to put them on a chart. Okay? And they're going to group them based off of their, whatever chromosome they are. Okay? So here we have chromosome 1. Okay? And so we see the 2 images of the chromosomes being placed here. And we see the 2 chromosome 2s in the cell. We see the 2 chromosome 3s in this cell, 2 chromosome 4s, and so on and so forth. Okay? So you're going to be able to see the numbers of chromosomes happening within each particular cell. Okay? Now, when we look at the number of chromosomes in each cell, we call it the karyotype. And so the karyotype is just the number and the appearance of each of the chromosomes within each cell of the body. Okay? So again, you should have 2 sets of each chromosome, when you're after meiosis, when you're starting to replicate your cells and forming more diploid cells. Okay. Now, there are ways that we can end up with errors in the chromosome number or disorder in our chromosome number. Okay. This happens through a process called non-disjunction. Okay. So non-disjunction is a scenario where the homologous chromosomes or the sister chromatids do not separate properly, either during meiosis 1 or meiosis 2. Okay? And so we're going to take a look at what these 2 processes will look like in each of these stages here. Okay? And so the risk of non-disjunction typically increases as parents get older. Okay? So the older the parents become, when they start having kids, the risk of non-disjunction increases. Okay? So we're going to take a look at what this will look like here. Okay? So non-disjunction can happen in meiosis 1 or in meiosis 2. Okay? And so if it's happening in meiosis 1, what happens is that chromosomes, the homologous chromosomes, do not separate properly. Okay? So kind of if we look at a normal meiotic division, here we should have our chromosomes lined up. Okay? Their homologous chromosomes lined up in their tetrads. Okay? They go through meiosis 1, and we end up pulling each individual chromosome to opposite poles of the cell, breaking the chiasma. And then they go through meiosis 2, everything's fine, and then you end up with your gametes at the end, okay, where you have 2 sets of chromosomes at each in each cell, okay, or a set of chromosomes in each cell. Okay. If we have non-disjunction in meiosis 1, okay, what happens here is the chromosomes do not attach to the mitotic spindles properly. Okay, so here this chromosome might attach properly, this one might attach properly, but for example, this tetrad, okay, doesn't attach to the mitotic spindle properly. It only attaches at one side. Okay. Now, because it only attaches at one side and they're still attached to each other at the chiasma, you end up pulling both chromosomes to one end of the cell. Okay. And so after this whole process happens, you end up forming gametes where you have 3 chromosomes in 2 cells, and then in 2 cells you only have 1 set of chromosomes. Or sorry, let me backtrack. Let me say that differently. You have an extra chromosome in 2 of your daughter cells and 1 missing chromosome in 2 of the daughter cells as well. Okay. And again, that's going to be due to pulling too many chromosomes to one end of the cell and too few chromosomes to the other end of the cell. And so you end up with 3 sister chromatids here in meiosis 2, and then 1 sister chromatid here in and the other cell in meiosis 2 here. Okay. So you end up having gametes with that are either missing a chromosome or they have too many chromosomes. So Paul's question is this how someone would end up with Down syndrome? Yes, this is exactly what happens in Down syndrome. Okay? And we'll talk about that in a second too. Okay. So this is non-disjunction of meiosis 1. Does this make sense so far? If this makes sense, go ahead and throw a check mark or a heart or something. If it doesn't make sense, go ahead and throw an X, and I'll explain this again. We feel okay with this. Okay. All right. Looks like we're doing okay here then. Okay. All righty, cool. So that's meiosis 1. So that's non-disjunction in meiosis 1. Okay. Now, if we're looking at non-disjunction in meiosis 2, okay, everything happens properly in meiosis 1 here. Okay, we still separate our chromosomes at the chiasma. They end up pulling to opposite ends of the cell. Okay, but then when they get to meiosis 2, the sister chromatids don't separate properly. Okay, so again, the mitotic spindles don't attach properly at the kinetochores. Okay, and so because they don't attach properly, you end up pulling a full chromosome to one end of the cell. Okay, and so at the end of this process, okay, here you have a chromosome that has one too many chromosomes. Okay, that's gained a chromosome essentially. You have one that's missing a chromosome over here. Okay, and then you have two gametes that end up having the normal amount of chromosomes. Okay, so one's missing a chromosome, one has an extra chromosome, and then two chromosomes are built properly. Okay, and so meiosis 2 shows you kind of the importance of that M checkpoint that we looked at in mitosis last week. Okay, so the M checkpoint, like we talked about, is important to make sure that all of the chromosomes have attached to the mitotic spindles properly. Okay, if those mitotic spindles do not attach properly, let's say we push past the M checkpoint before those mitotic spindles are attached to the chromosomes properly, okay, then you end up pulling an uneven number of cells or an uneven number of chromosomes to opposite ends of the cell. Okay, and then you end up with non-disjunction here. Okay, and so you end up with an inappropriate amount of chromosomes in some of your cells, in some or all of your cells, depending on where this non-disjunction takes place. Okay, cool. Are any questions on non-disjunction so far? Okay, cool. Okay. So non-disjunction will typically happen with the non-sex chromosomes. So chromosomes 1, so remember we have 23 pairs of chromosomes, right? We have chromosomes 1 through 22, which are considered the autosomes. Those are the non-sex chromosomes. Okay? And then we have the X and the Y chromosome, which are considered our sex chromosomes. Okay? So the non-sex chromosomes are referred to as autosomes. Okay? So an individual who has the appropriate number of chromosomes, or has a normal amount of chromosomes in their cells, is considered euploid. Okay? Euploid, eu meaning true, ploid referring to chromosomes. So euploid has the correct number of chromosomes in these cells. Okay? 2 sets of chromosomes. Okay? Someone who has an error, okay, someone who has either one too many chromosomes or one fewer chromosomes, will be considered aneuploid. Okay? So that's where you have an error in the chromosome number. Okay? So the most common aneuploidies are monosomies, where there's a loss of a chromosome, so you only have one copy of each essential chromosome, okay? Or a trisomy. Okay? And so a trisomy means you've gained an extra chromosome. So instead of having 2 chromosomes, you now have 3. Okay? Trisomy tends to be a little bit more dangerous depending on where that chromosome or where that trisomy occurs, okay? Depending on the chromosome that has trisomy in it. Okay? And so sometimes the lifespan can vary. Sometimes people will be able to live a healthy life with the trisomy. Sometimes they won't even make it to term if they're developing within the womb. Okay, and so the reason for that is because of what's called gene dosing. Okay? There's an imbalance in gene dosing. Okay, so remember, genes are going to be responsible for building the proteins in order to allow that organism to, or allow that cell to function. Right? If you have one too many chromosomes, you're going to be producing too much of that particular protein because you have 3 genes potentially being activated instead of just potentially 2. Okay, so because you're producing extra protein, okay, it creates an imbalance within the cell, and that causes the cell to kind of go out of whack, and it causes some significant issues. Okay, so again, in some cases, this gene dosing can be lethal. Okay, lethal very early, and in some cases, you're able to still live throughout life with a trisomy. Okay. So an example of a condition that has non-disjunction, like Paul mentioned, is Down syndrome. Okay. So Down syndrome is where you have a trisomy of chromosome 21. Okay. So instead of having 2 sets of chromosome 21, okay, you end up with a 3rd chromosome. Okay. And so people who have Down syndrome, and you don't need to write down the slide, I'll tell you which slides you don't need to write down. So we're going to look at various conditions. The condition slides you don't really need to write down, just worry about the actual material part. Okay. So don't worry about writing down the slide, but this just is to kind of help, put this into context here. Okay. So when you have a trisomy of chromosome 21, you end up with 3 chromosome 21s due to non-disjunction. Okay. And so people who have Down syndrome will usually have delayed growth. It takes them longer to grow, and then delayed mental capacities as well. Okay. And then they'll start to take on some pretty characteristic, usually facial characteristics that are usually associated with Down syndrome. And then the kind of prevalence of non-disjunction, or the likelihood of having someone give birth to someone with Down syndrome typically increases as the mother gets older. Okay, so if a mother is in her 20s, the chances of her having a baby with Down syndrome is probably less than around maybe 0.1 or 0.2% or so. Okay. As they start to get older, okay, the likelihood starts to increase. And so once they hit about age 40, okay, now there's a 0.75 chance of having a baby with Down syndrome. Okay, still that's a 99.25% chance that you won't, but you can see the likelihood increases dramatically. Okay. And then once you hit age 45, okay, now you're looking at almost a 4% chance of having a baby with Down syndrome. Okay, so again, you still have a 96% chance of not having a baby with Down syndrome. But you could see the risk increases dramatically when you're in the mid 40s compared to a mother who's giving birth in her 20s, okay, 20s or 30s. Okay, so it's still unlikely, but it's still the incidence does increase dramatically here. Cool.
Is there a way
that's
to prevent the extra chromosome?
Not really. Yeah, so his question was, is there a way to prevent that extra chromosome from developing or having that non-disjunction occur? There's nothing really you can do because you don't know that it will happen. And even if you did, there's not really anything, there's no tools to kind of make sure that non-disjunction doesn't happen. It either happens or it doesn't. So there's really not much you can do at that point. Yeah, you just have to kind of roll with the punches and see what happens, unfortunately. Yeah. Okay. So that's one disorder of chromosome number. Oh, so Paul's question. So Paul has a question. So does the father have anything to do with it? Typically, no. So the father, his role is very limited in terms of non-disjunction happening here. In Down syndrome, it's usually just the mother who is the person in question here. Father, not so much. Cool. And I think part of the reason for that is because when it comes to males, they're able to produce sperm and they're able to, it typically works fine. However, when you're dealing with the woman, things start to wear out because the ovaries have already held on to so many eggs for long periods of time. The longer they wait, the more damage they can kind of incur, and so that can kind of lead to some of these processes happening here. So the next type of disorder happens with what's called X inactivation. Okay, so this happens specifically with the X chromosomes. Okay, so if we remember when we're looking at females, okay, females are going to be they're going to have 2 X chromosomes, while males are going to have an X chromosome and a Y chromosome. Okay, so like we talked about before, monosomies and trisomies can be very detrimental. It can be sometimes lethal, okay, due to the imbalances that are caused by gene balancing or gene dosing. If there's excess gene dosing or not enough gene dosing, that can cause imbalances in the proteins that you're able to produce. Okay. Now, when we look at males and females, we're going to see a different number of X chromosomes. So, like we said, females are going to have 2 X chromosomes, while males will only have 1. However, females and males are both going to be able to function normally. Okay, they're both going to be able to work properly. Okay, and so this is going to be kind of curious. Like, why are males able to work properly with just 1 X chromosome, while females are able to work properly by having 2 X chromosomes? Okay, so we're going to take a look at that. And so for females, in order to produce a female, the female has to inherit an X chromosome from both their mother and their father in order to produce a daughter here. Okay. In the case of males, males have to inherit a Y chromosome from their dad and then an X chromosome from their mom. Okay. Now, when we're going to be focusing mainly on females here. Okay, so females, are going to have an extra X chromosome. Okay, so instead of having one X chromosome, they're going to have two. Okay, however, they're still going to be able to function normally, right? And so the reason they're able to function normally is because of what's called X inactivation. Okay? So essentially what happens is when a woman is developing inside her mom's womb, okay, when you have a daughter developing in a womb here, okay, one of those X chromosomes that she has produced is going to be inactivated, okay? And it's going to condense into a structure called a bar body. Okay? Essentially what the bar body is just a condensed chromosome that doesn't do anything at that point anymore. So here again, we have two X chromosomes. Okay? At a certain point, one of these chromosomes condenses into our bar body and inactivates. Okay? And so you only end up with one X chromosome being fully activated here. Okay? And what that does is that prevents excess gene dosing. It prevents the X chromosome from producing too much protein here. Okay? Again, because you only have one X chromosome that's active. Okay? And so this prevents excess gene dosing. And so only the genes on the activated X chromosome are going to be expressed. Okay. Now, kind of an interesting kind of thing here is that if a cell has a maternal X chromosome that gets inactivated, then all of the cells that come from that cell, all the daughter cells that come from that cell will also have that maternal X chromosome inactivated as well. Okay. So whichever X chromosome gets inactivated, from that cell, that's going to be the one that gets inactivated from that entire progeny of cells. Okay? Same thing as if it were the paternal X chromosome. Okay? So if that paternal X chromosome gets inactivated, then all the progeny of daughter cells that come from that initial cell will all have that paternal X chromosome inactivated as well. Okay. So that is X inactivation. Essentially, one of the X chromosomes in females becomes inactivated to prevent any excess gene dosing here.
So I know this isn't related, but I remember you said that for males, yeah, the Y chromosome, it's shrinking, and and there's a theory that at one point it will cease to exist. If it continues to shrink, like when will that point be? Like, will that be like millions of years or like a couple hundred or what?
Yes, that's a good question. I'm not sure off the top of my head. I'd have to look into it again, but my guess is it would probably have to be over a couple hundred years because this process is extremely slow. It has been decreasing over multiple years or shrinking, but it would take a while before that would actually happen. Yeah.
Okay.
Cool. Yeah, good question. Okay, cool. All right, so that's X inactivation here. Okay. So when we're looking at X inactivation, there can be certain scenarios where an individual that has an abnormal number of X chromosomes, they can end up with having, for example, a trisomy of X chromosomes. So maybe they have 3 X chromosomes or have 1 X chromosome. Doesn't matter how many chromosomes that they produce, okay, all but one of them is going to be inactivated anyway. Okay? So there will always be one active X chromosome, okay, regardless of if there's a trisomy in it. Okay? Now, when there is an abnormality in that X chromosome, that is going to lead to various types of defects. Okay? Sometimes it can lead to mental defects, sometimes it can lead to physical defects, and sometimes it can even lead, and most of the time it's actually going to lead to sterility, which means that you're not going to be able to produce any offspring. The reason for that is, like I mentioned, I think it was last week, the X chromosome has a lot of really, really important genes that are important for life. Okay? So if there's an abnormality in the X chromosome, that causes a lot of different things to kind of go out of whack and then create some significant health issues. Okay. And in some cases, there can be certain scenarios where the X chromosome doesn't develop altogether. If that happens, then that individual will not make it to term, while they're developing within the uterus. You have to have that X chromosome in order to live. Okay? If you don't have an X chromosome, you're not going to be able to live. You're not going to be able to even be. You're not even going to be able to develop within the womb. Okay. So we're going to look at a few of these conditions. At this point, I'll tell you what you need to write and what you don't need to write. For now, don't write anything because this is not going to be important for the exam, but this is just a few examples of some conditions that can happen here. Okay. So this first condition is called triple X syndrome. And so this happens where you have a trisomy of chromosome 3, the X chromosome. So you have 3 copies of the X chromosome here. Okay, and so with 3 copies of the X chromosome, okay, you don't have any Y chromosome, so these individuals end up becoming female. Okay. However, because they have an additional X chromosome, this causes a lot of developmental delays, and so they don't develop within the womb properly, and then even when they are born, it takes them a lot longer to develop over the normal lifespan. Okay. And then they also have reduced fertility as well. Okay. And depending on person to person, the severity of it can vary. So individuals who have the triple X syndrome typically are very tall compared to the average woman. And this happens in about 1 in 1,000 births here. Okay. So again, that's going to be due to a trisomy of of the X chromosome here. Okay. Another example is what's called Klinefelter syndrome. Okay. So in Klinefelter syndrome, they have a genotype, or they have 2 X chromosomes and a Y chromosome. So because they have a Y chromosome, they are inherently male. However, because they have an additional X chromosome, they start to develop a lot of kind of more female characteristics here. Okay. So people who are dealing with Klinefelter syndrome will be male, but they will have very small testes, and they'll start to develop some breasts. Okay. And they'll also have reduced body hair as well. Okay. So even though they have 2 of those X chromosomes, that extra X chromosome does become inactivated, but its presence alone causes these other issues to start to arise. Okay. And so this will happen in anywhere from 1 to 500 to 1,000 newborn males. The next one is Turner syndrome. So Turner syndrome is where you have only a single X chromosome and nothing else. Okay. So it's given the genotype X0. So it only has 1 X chromosome. There's no 2 X chromosomes, and there's no Y chromosome. Okay. So because there's an X chromosome, these individuals are female. Okay. And this happens in about 1 in 2,500 newborn children. And so this usually will start to, the characteristics for Turner syndrome will usually start to arise around age 5. And so these typically will be females that are very short. They have a lot of really webbed skin around their neck region. Their chest is very broad compared to normal. And then a lot of times they'll have lots of hearing and cardiac problems as well. Okay. And then they'll develop some sterility, and they are not able to produce any offspring because of issues with their ovaries. Okay. So the ovaries don't function properly. Okay. And so they end up not being able to have any children. Okay. They end up being sterile. Okay. And so in order for them to kind of go through puberty, they have to go through hormone therapy in order to kind of guide them, guide them through the stages of puberty. Yeah. So Mario's question, so do they also look very childlike? So in some cases, yes. However, this is just I'm not sure how old this individual was here. Without hormones. So yeah, so without the hormones, they would essentially look very childlike. So they wouldn't develop, they wouldn't develop breasts. They wouldn't develop, the natural kind of processes that happen during puberty, because they're going to need those hormones in order to help them have those changes during puberty. If they're not developing those hormones, they're not going to be able to do so. Okay. So what the hormone therapy does is it kind of helps them become, kind of take them out of that, that childlike form, and kind of helps them kind of develop further. But it takes a little while before that can actually take place. Yeah. Good question, Mario. Any other questions with any of these diseases here or disorders? Okay. Okay. So when we are looking at conditions where we have errors in the chromosome number, we call that polyploidy. And so polyploidy is where you have more than the correct number of chromosomes. So usually that's going to be triploidy. So in triploidy, instead of having 2 sets of chromosomes, you end up with a set of 3. Okay, 3 sets of chromosomes all the way across the board here. Okay. So chromosome 1 has 3 chromosomes, chromosome 2 has 3 chromosomes, chromosome 3, there's 3 of them, so on and so forth, all the way across the board. Okay. And so the way this will typically happen is, instead of producing an egg that is haploid, like normal, a woman might release an egg that's diploid. Okay. And so when that egg is diploid, it fuses with a haploid sperm, and so now you end up with 3 sets of chromosomes across the board here. And so you end up with a triploid zygote. Okay? And so triploids or triploid zygotes are typically sterile and a lot of times won't make it to term just because of excess gene dosing here. And so these ones are pretty lethal. If this happens in a baby that's developing in uterus, that baby most likely won't make it to term. Okay? And so if they are able to make it to term, there are some organisms that are able to survive as being a triploid organism, but they're not able to produce or not able to undergo meiosis and produce sperm cells and egg cells because they have an odd number of chromosomes. Okay? So for meiosis to take place, you need an even number of chromosome sets. If you have a triplet organism, you're going to have 3, you're going to have an odd number. You're not going to be able to produce any sperm cells or egg cells properly. And so they're going to become sterile. Okay. So that's errors in chromosome number. Okay. So Paul's question says, so is this what happens with a liger? Yeah, so this is essentially what happens with ligers. So when tigers and lions kind of mate, they are able to form a zygote, which is an offspring, which would be the liger. Okay, but because the liger doesn't have the proper number of chromosomes, and because of other issues that happen within the liger itself too, it's not able to produce any offspring from there. Yeah, really good, really good. Cool. Okay, last one here, as we kind of are running out of time here, is disorders in chromosome structure. Okay, so disorders in the chromosome structure have to do with the chromosomes just not being built properly. Okay, and so we can have different types. We can have what's called a partial duplication, where you actually duplicate a part of the chromosome. Okay, instead of just having it be existing as it is. Okay, you can have an insertion where you insert a part of one chromosome into a part of another chromosome that are not corresponding with one another. Okay, so for example, here we have chromosome 4 taking a chunk and adding it to chromosome 20. So that would be an insertion. We can have a deletion where you have a chunk of your chromosome getting removed altogether, like we see down here at the bottom. Okay, or you can have a translocation. So a translocation would be where pieces of different chromosomes end up getting swapped with one another. Okay, and so we'll take a look at these in a little bit of detail here. Okay, so whoops. So the first type of structural problem or structural disorder is called deletion. Okay, and so one of the conditions that has to do with chromosome deletion is called Cri-du-Chat syndrome. Okay, and so Cri-du-Chat syndrome is where you have a deletion of the smaller arm of chromosome 5. Okay, and so instead of having your chromosomes looking like this, where you have 2 healthy looking chromosomes, on one of the arms, on one of the chromosomes, you end up deleting a chunk of it, and you end up removing it altogether. And so people who have Cri-du-Chat syndrome will typically have a lot of nervous system abnormalities. They'll have very distinctive facial features. They'll have a relatively small head in comparison to their body and very weak muscle tone. Okay, so there's a lot of kind of issues that kind of arise because of the deletions, because you're removing large chunks of genes that are important for producing various types of characteristics. The next one is an inversion. Inversion here. Okay. And so inversions, you can go ahead and write the slide down. So inversions are where we have chunks of the chromosome getting detached, rotated, and then plugged back into the chromosome. Okay. So here we see chromosome, this chromosome here, we see gene B, gene C, and gene D. Okay. If you remove this entire thing out, okay, turn it around and then plug it back in, you end up with what's called an inversion. Okay, all you do is just invert the genome. Okay. Now, depending on the scenario, this can either be mild, producing mild effects, or sometimes it can be very detrimental. Okay, if you're removing entire genes, okay, and you're just rotating them and plugging them back in, okay, this is typically fine because your entire complement of genes is still there. Okay, so everything is still okay. You're still going to have might have a few mild effects, but everything is still going to be fine typically. However, if, for example, we decided to cut gene B in half, and then we rotated this portion here. Okay, we took half of gene B and then took all of gene C and gene D and then rotated it and then plugged it back in. That would create some detrimental effects because now you're having half of the genome or half of the gene of B. Okay, now in contact with D, and then C, and then the other half of B. Okay, so you reorient and you kind of disrupt the genes that you're producing, and so that can create some significant health issues. Okay, that would be very detrimental to the cell, and most likely detrimental to the organism as a whole as well. Cool. So that is an inversion. Okay. And the last one here is a translocation. Okay, and so a translocation is where you take a segment of the chromosome, okay, you remove it, and you reattach it to another chromosome. Okay, and so typically what will happen, or one type of translocation that we can have is called a reciprocal translocation, okay, which means that basically non-corresponding chromosomes, non-homologous chromosomes, will exchange some of their DNA with one another. Okay, so for example, here we have one chromosome, this blue chromosome here, we have this orange chromosome over here, okay? And so in a translocation, you would swap chunks of your chromosome. Okay? So you take a chunk of this blue and add it to the orange, and you take a bunch of the orange and add it to the blue. Okay? And so again, this is going to be between non-homologous chromosomes. So if you were looking at chromosome 4 and chromosome 20, for example, okay, and you swapped the parts of their genome or parts of their chromosomes, okay, that would be what's called a reciprocal translocation, because you're exchanging it from one chromosome to the next. Okay. There is typically no net loss of DNA, because the DNA is all still there, but it's just the chromosomes are not built as appropriately as they should, okay? Because now they have chunks of chromosome that shouldn't be there. And so this is a type of disorder that is usually associated with lots of different types of cancers, as well as schizophrenia as well. And that is it. Those are the different types of issues that can happen when meiosis does not work properly. Okay. So I will go ahead and leave it there. That's all we have for today. There is some classwork that's up on Canvas as well. So go ahead and do that. Make sure you complete that by tonight. There's a homework assignment that will be due when we come back from spring break on, I believe it's April 14th. Are there any questions before we take off for today?